Hello students, so in this video we will be continuing with the mechanism of transcription. Now in the last video we studied about the transcription unit and the things required for transcription, that is your promoter, sigma factor, terminator and rho factor. Now students, in this picture you can see the various regions, that is the promoter, the initiation site where RNA synthesis will begin, and the terminator region where the RNA synthesis will stop, the coding sequence and the template sequence. The template sequence will be transcribed. Okay, now the region which is close to 5 prime is known as the upstream, and the region which is close to 3 prime is known as the downstream region. So here we can say that the promoter region lies at the upstream region of the coding sequence, and the terminator region lies at the downstream of the coding sequence. Now from here the RNA synthesis will begin, that is RNA polymerase will come and bind to the promoter region, that is the initiation site, and in binding of the RNA polymerase to promoter region the sigma factor will help. Now students, in this video you can see the RNA polymerase is being helped by the sigma factor to get attached to the promoter region. After the RNA polymerase gets attached, the sigma factor falls off. Then the double strand unwinds, breaking the hydrogen bond between them, and RNA polymerase can start synthesizing the mRNA by adding nucleotides. Now in this picture you can see students, elongation has started, that is RNA polymerase started synthesizing the RNA, and this will continue unless and until the termination site appears. As soon as RNA polymerase comes in contact with the termination site, the RNA synthesis will stop. The rho factor will get attached to the RNA polymerase and helps the RNA polymerase to stop synthesizing the mRNA. Now as soon as the RNA synthesis stops, the new RNA, the enzyme RNA polymerase and the rho factor falls off and the double-stranded DNA rewinds. Here you must remember students that RNA synthesis will be from 5 prime to 3 prime direction.